Yeah. Oh, I got you to 3.
All right. Everybody signed in that's going to be signed in. George, I know we just started. If you want to go ahead and get signed in, I don't know if you've gotten in yet. I'll give you a second.
I need the color. Blue. Blue helps you remember stuff. Don't know why it does. When you write, like I write so hard. No, me too. And then when I reset, it's still clear. Oh, I'm like, my paper is good for something. I just use these $2
You do this. Yeah, that's how you get it. That's what I'll always take attendance from. Yeah, because we haven't started yet. So. Yeah. It did remind me that I do have that, but only for people that can't do this part.
I'm currently in the process.
Yeah, we're running late because they replaced the whole computer and I had to like redo everything on it. Like normally I'd come in here and start the computer and it's ready to go, but
Hey, Professor Yost, should I join since I'm in the...
George, all right, Spencer, I'll let you get signed in real quick. Then we're gonna get going.
Oh, I need to make a microbiology grouping. I need computer remind me to do that. Spencer, can you message me over to remind me to make the microbiology one? A group is basically a big group chat so we can all help each other. And I
all right, let's go ahead and get started. This is going to be review over chapter 2. All right, which type of chemical bonds involve sharing electrons? All right, so that's going to be covalent. Good job, guys. Most of you guys got that one. So sharing electrons is covalent. What about hydrogen bonds? Pick the answer that best describes this. All right. So the answer for this one is going to be hold water molecules to one another. Now, most of us picked the green one, form between hydrogen atoms in different molecules. That should be obvious to you guys why that's wrong. Because when we talk about hydrogen bonds forming, we're talking about a polar hydrogen. So when a hydrogen is polar, what polarity does it have? Positive or negative? Always positive. Always positive if it's polar. Okay? Is positive going to be a charge going to be attracted to another positive?
No.
Guys. Okay. So what is positive going to be attracted to?
Negative.
Negative. Right. And so where do we see that happening? Between water molecules. So between the oxygens, which have our negative polarity, and our hydrogens, which have our positive polarity. You're not going to see hydrogen bonds forming between hydrogens because positives aren't going to be attracted to positives. And if hydrogens aren't positively polar, then they are non-polar. And non-polars aren't attracted to things. And so they're not forming hydrogen bonds. Hydrogen bonds, yes, you have to have at least one of the atoms be a hydrogen, but that means the other atom is a non-hydrogen, and that means it's also going to have a negative polarity, the other atom will. Okay? So that is all defined within our understanding of what's actually going on with our electronegative and our, well, our electronegative, like oxygen, it's usually oxygen. So. And our hydrogen as our positive polarity. So keep that in mind with our hydrogen bonds. It's not between two hydrogens, it's between a hydrogen and something else. All right, next question. What element is in all organic molecules? Should be an easy one for you guys. This is loud, man. All right, carbon. That one's pretty straightforward one. Most of us know this one. If it's organic, it has to have a carbon backbone. Usually, if we're calling it organic, it's going to have hydrogen attached to carbons as well. All right, true or false? Water is a poor solvent. Is that true or false? All righty. So false, yeah. And why is water a good solvent? Because of that polarity, right? The oxygen's being negative polarity, the hydrogen's being positive polarity. All right, what is water's pH? Pure water. You guys know this one. All right, good job, guys.
It's like completely in the middle.
Because it's completely in the middle, it's completely neutral. Exactly. So what range on the pH scale would an acid be? Okay, I'm glad that you guys are getting that stuff. At least the pH part won't be too bad for you guys. All right, lipids. Which are the choices? Best applies to a lipid. I already 10 seconds. Should I give 20? I don't know why I put your last name up there, Lily, but you got a last name. All right, so lipids, they can be used to make energy for the cell. We know this because your body stores fat literally for that purpose, like later on. It can be used to make hormones. That's what we talk about with the sterols. And it can be used to make the cell membrane, like our phospholipids. So good job, you guys got that one, all of the above. All right, which of the following organic molecules is amphipathic? Guys, think about what amphipathic means. All right, so that's our phospholipid. Remember, amphipathic means we have both going on. That means we're going to have a polar region and a non-polar. Now, nucleotides, now those are going to consist of a sugar, a phosphate, and the base. The bases, are they, now, and I know we didn't talk about this specifically, but if we think about our nitrogenous bases, what makes it A, T, C, or G, right? Those nitrogenous bases on there on our nucleotides. Are they polar or are they non-polar? Do you guys know?
I believe they're non-polar.
They are polar. You want to know how we know that? Because they make the hydrogen bonds in the middle that hold your DNA together. So even that deductive reasoning, we should already be thinking about that going on. Even if we didn't say it directly, we did talk about hydrogen bonds between DNA. The bases are in the middle. That's what holds that DNA together, right? So glucose, the sugars, we didn't really talk about that too much, but we should, most of us be aware that sugar dissolves pretty well in water, probably polar, right? Amino acids, those can kind of vary. It just depends on the amino acid, all right? And then for the phospholipids, those are always going to have the charged head and the hydrophobic tail. Yeah.
Isn't the phosphate positive while the tail is negative?
Phosphate's actually negative, and the tail is hydrophobic or non-polar. All right, so what is a monomer of a nucleic acid? So what was the term that we used for that? We'll kind of just talk about that a little bit. All right, so that's a nucleotide. Nucleotide is the monomer for nucleic acids. Those are repeating units. Yes, a nucleotide can be broken down further to a pentose sugar, the phosphate, and then that nitrogenous base. We're going to talk about that a lot in chapter 6. So it's just our primer for that stuff. Alpha helices and beta pleated sheets are examples of which type of protein structure? Yeah, you get 30 seconds for this one for some reason. Whatever.
I know I know this one.
You guys, this is one of those moments where like, I had coffee this morning and I had my mask on and it smelled like coffee breath. So I'm like, we'll chew some gum. You guys ever chew gum with a mask on? Like the mintiness will like get into your eye. That has the same effect as like Vicks Vapo Rub or something. Yeah.
However, if you're dealing with a C. diff patient,
Helps.
Vicks Vapo Rub
Yeah.
in your first mask, put a mask over that one.
Yeah, that's what I've heard.
It's great.
Yeah. Also good with cadavers and things like that if you guys
I
end
have
up doing that stuff.
I have 12 family members in nursing, and
Yeah.
every single one of them said C. diff, get the Vapo.
Yeah. All right, good job, guys. Remember, this is participation based, so if you didn't get them all right, don't beat yourself up. Just get you an idea of how to start thinking about this stuff and start applying it. If you guys didn't see, I posted the critical thinking questions, some examples. I gave you 5 examples of critical thinking type questions that might appear on the exam. They're pretty good representations of what those questions will be like on the exam. They will not be the same questions as what's on the exam, okay? Don't bank on that. If you are going to look at those critical, when you're ready, again, I would recommend at least getting through the content before you challenge yourself with those. But when you're ready to start breaking those questions down for yourself, don't just look for the right answer. Also, if you can start thinking about why the wrong answers are the wrong answers. So we should always be assessing our questions. Like first, read your question. What do you think the answer is going to be? And then go through all of your choices. Don't ever just say, oh, this looks like the right one and not read the other choices. That's a pitfall that too many people fall into. Make sure you read all of your choices before you finalize your answers on the exams at the very least, right? Okay. Thank you guys for bearing with me on that. Good job. All right, modules. Bear with me while I download the slides. All right. Week 1. Week 2 is what we want. Slides.
I just like how you went, go to a pig barn. A pig barn. Their urine very high in pneumonia. Trust me, I used to own pigs, and there was this thing called pig show fever, which was a very minor case of pneumonia overdose, where you would vomit profusely.
All right, good news. This computer doesn't have PowerPoint on it. So we're going to be viewing this in Acrobat, I guess.
Professor Yode's crap.
Yeah, IT is going to be hearing from me after this. You better believe it.
And mono? My sister got water. Very sick. It ruined your lungs. No, it can't. It really can't just let. Which is good. I had bronchitis as a kid. Yeah. My lungs have never been the same.
Oh my gosh. So anyways, let's just do it from here, shall we? All right, so we're going to talk about bacteria and archaea versus eukaryotes. We're going to introduce the concept of what eukaryotes are versus akaryotes. Other term for akaryotes is prokaryotes. Remember, akaryote means without, like you have asymptomatic. 1/3 of all COVID people who have COVID, like would test positive on a COVID test even. 1/3 of the people that have COVID are asymptomatic. They don't have symptoms, which is part of the problem of controlling that, because everybody just thought, I don't have symptoms, I must not have COVID. Incorrect. You could have it right now while you're sitting here feeling just fine. So 1/3 of people don't have symptoms, asymptomatics without symptoms. So eukaryote without karyote, and we just have to know that means nucleus. So bacteria and archaea, they do not have a nucleus. If they don't have a nucleus, they also don't have any other membrane-bound organelles. These are parts of the cell like mitochondria and a plastic reticulum, which we'll talk about in chapter 5. These bacteria and archaea do not have any of those membrane-bound organelles. right, I can't do that because this is stupid. All right, so the bacterial cell structure, this is a good place to come back to. I'm not gonna go over it individually now, obviously, but this is a good place to come back to for your studying. You can zoom in on this pretty well when you get it on PowerPoint, or if you get it on PDF, you'll be able to look at this and zoom in on these and help you guys study this different structures of bacterial cells. Because even though they don't have membrane-bound organelles, they still have quite a lot going on with them, right? Right. These are single-celled organisms, always. Bacteria and archaea are always single-celled organisms, okay? They are cells that are capable of themselves reproducing. They have their own metabolism, so they have breaking down and making of chemical bonds and processing of nutrients. They can act as a group in colonies where they will literally like just multiply up and pile up on one another, or in biofilms. They have the ability to signal between each other with waste products and other chemicals they can produce that notify the cells nearby them what's going on. As far as nanowires go, I don't know anything about it, and I'm not going to ask you about it on the test. All right, we have 3 general shapes of bacteria: the coccus, the bacillus, and curved. So curved just refers to anyone that don't have the rod shape, which is our bacillus looks like a pill or a rod, and then the coccus is our round ones. So just typically what we're really talking about. I really want you to focus on coccus being round and bacillus being rod. Okay. So think of coccus, the C's, all the C's and the O's, that looks round to me, mind you being round. And the bacillus has those L's that look like rods already. So that's our rod shape. Okay, so that helps you remember which one's which. If it's got a spiral or a curve to it, it falls into the curved category. I feel like spirillum and spirochete, those guys kind of give it away that they're spiral shaped guys. And I'm not going to ask you about the difference between those or anything, but do know that those don't fall into coccus or bacillus. Okay, so they're going to be curved. Pleomorphism, this means that this particular species of bacteria doesn't have a set shape. It can have a variety of different shapes. There's certain kinds of bacteria that fall into that area, and we'll talk about them in a minute. All right, so here's some examples of our shapes and our arrangements. Here we have in our coccus, which we get round, right? We have our C's and our O's, reminds us of round. See how round these cells are? See how round these cells, these individual little balls are? So those are our actual shapes. We're going to talk about the arrangements as well. For a rod, guys, you see how these are definitely rod versus the actual round shapes of these guys. These guys have more of a curve. I'm never going to give you a picture of this curve versus these rods and tell me the difference, okay? I'm never going to do that to you. In fact, 99.9% of the time on your test, if I ask you about cell shape, it's going to be coccus versus rod, or the answer is going to be coccus or rod. I'm usually not going to ask you about the other shapes. Okay, our bacteria in our lab, I only have cocci and rods. I have one species that's a coccobacillus. It's like all oval shape. And if you guys get that one, then I'll let you know that ahead of time that, hey, you're going to write down coccus or you're going to write down bacillus. This one just happens to be coccobacillus. So for our arrangements, cocci have specific arrangements they can have, and bacilli have different arrangements that they can have. For our cocci, obviously we can have diplo, that's just pairs. Tetrads, groups of 4. Then we have staphylo and strepto and sarcina. And sarcina is a cube. Okay, so it's going to have equal numbers, like literally cubed of whatever numbers in these stacks. And you don't see that a lot. But the big ones that I definitely want you to be aware of, you should know diplo and tetrad just because of the words kind of give away what they mean. So I'm not going to focus on that a lot. But staphylo and strepto, I know you've heard of both of these. Strepto to me sounds like strip. It's chains. Yeah, like strep throat. So that sounds like strip to me, which these are chains. These are linked together. So we go back up to this image that was up here. See how these guys are linked together in long chains? Guess what? That's strepto. And in fact, this guy is named Streptococcus pyogenes. So streptococcus tells you right there in the name, strepto, it's going to be strips, chains of coccus, cocci, round cells. So that name gives it away, okay? Same thing with the one above it, staphylococcus. So if we come down here and we look at, be sure I'm not missing anything on mine. We look at staphylococci, and I'm not going to ask you about micrococci, but staphylo means irregular or grape-like clusters. And so when we look at this picture of staphylococcus aureus, you see how they're kind of bunched together in random bunches, but they're not all over the place. They're clearly like bunching up. And you'll even see that when we look at them in our slides in the lab. So Streptococcus will mean in chains of cocci. Staphylococcus, we have random bunchings of cocci, okay, round cells. So there's in their names in those. So Staphylococcus aureus is going to be random bunches of round cells. The aureus part is giving you that specific epithet, so we know what species it is. Just so happens that aureus means golden glow. Their colonies have a golden color to them. So that's why it's named that. So it actually has a lot of descriptors just in the name. However, we go to something like Escherichia coli. That's not giving you any information about the arrangements or the shapes. So not all of them do give that away, but some of them were lucky enough, and then you can make inference based on that, because if they have it in their name, they follow that rule, okay? Like here, we have Legionella pneumophila. Does that tell you it's gonna be rod-shaped when we said rod is bacillus? It doesn't tell you, does it? Look at streptobacillus. Again, still strips or chains of bacilli. They're just linked together end to end. So that has it in its name. All right, so moving on to the bacilli, we already talked about streptobacilli, diplo, again, pairs, okay? There's one other arrangement that these bacilli can have, it's called palisades. This is easy to imagine if I were to talk about this, and we were to talk about palisades being like fence posts. They're literally like the rods are like up next to each other. Typically they're linked at like one end or another. So you could imagine it being almost like zigzag lines, if you will. Sometimes they're linked at the top. Sometimes you might see them like this or whatever. But basically, if you were looking at them in a microscope, they're lined up like height-wise instead of end to end. Okay, so palisades, a palisade is literally a fence or a barrier. It's another word that we use outside of discussion of microbes, if you've ever heard of that term being used. So palisade is like a barrier. And so think of a fence with that one. All right. So we also talk about spirilla or spiros, S-P-I-R, think of spiral. Okay. They can be in chains or they can be next to each other. Close, I guess. Here we go. All right. Moving on to our biofilms. This is where we have multiple species of bacteria living in an environment together. They're often stuck together with some nice gooey stuff that they make. And again, they can signal each other with like certain waste products that they're making, or with certain literal proteins and other chemicals that they can make that signal the bacteria nearby that things are going good or bad. Obviously, if you're having a lot of products that are being made from dead cells, that's kind of showing you nearby, hey, the guys nearby are dying a lot. Maybe we shouldn't be associated with biofilm anymore. So these guys can actually communicate with each other with those chemical signals in a biofilm. On a good example of a biofilm, we're talking about the plaque on your teeth. It's a very good example of a biofilm. More than one bacteria involved in that. You can even have more than one type of organism. There could even be in your biofilm, archaea and bacteria, or even protozoa get involved in it. So, but we already talked about these guys mostly are going to have like a surface, something for them to stick to. And they will stick to it and make their own sticky coating to help them stick to it. And then they'll build up on top of that with that little sticky coating that they make. And then the bacteria can come in and stick onto the stickiness and send each other all these messages. And that communication is called quorum sensing. If you ever heard us talk, and we probably don't, I don't know why you guys are listening on the back of the Senate, but if you ever heard of like a Senate or Congress or whatever, talking about how they have quorum, that means that they have enough people there to actually do business, take votes and things like that. So quorum sensing is sort of like that, getting the feel for what the general population is doing. And so that's from those chemicals they're making to communicate with one another. Next, we have the types of appendages that can exist on bacterial cells. Now remember, they don't have membrane-bound structures inside of the cells, but they can have these appendages that can kind of stick out from the sides of the cells. There's two kinds of appendages, the ones that are used for motility and the ones that are used not for motility, okay? The only one that's used for motility is flagella. The other ones are fimbriae and pili and nanowires. These guys are usually for attachment or to create channels between each other. We're going to talk about them. All right, so flagella, these are appendages. You can see here, these little hair-like guys that are squiggling off the end of the cell. So these structures here, this broad shape, so that's bacillus. I've got these squiggly guys coming off the end of it. That's a flagellum. One is a flagellum, multiple is flagella. And they are almost like a motor. It's proteins, basically that do work sort of motor-like, and they rotate the flagellum, okay? So you can have more than one of these guys. You can have them all over the cell, or you could just have a couple. It just depends on the species that we're talking about. And some bacteria don't even have a flagella. They're non-modal. And so depending on if you have them, if you have a flagellum, you are a modal, and you might have more than one. These guys rotate one clockwise or counterclockwise, and that's how they control their movement. But they do not whip. Okay, it's very important. They don't whip, they only rotate, clockwise or counterclockwise. You can have several different arrangements around the cell. And here are our, obviously you could have no flagella, then just not have flagella and be non-modal. So we have polar arrangements and non-polar arrangements. Our polar arrangements, that's referring to if you have them at the ends or both ends, but if they're just at the ends or one end or the other, that is our polar arrangement. So just look at the poles of the cell, all right? But you could also be covered in them completely, which is peritrichous. So peritrichous means that you have flagella all over the cell. That's a non-polar arrangement, technically, because they're not having it one end or the other. So here we can see some images. So trich, if you ever heard of like trichotillomania or something like that, trich or trichology or things like that. That trick word, T-R-I-C-H, that means hair. So it's like a hair-like appendage. It is also made of protein, so that tracks. But you could have monotrichous, so you have one single flagellum. So that's our image in A. It's my phone, not mouse. Lophotrichous, you have bunches or tufts. That's C. This is lophotrichous here. So at one end of the cell, you have a tuft of flagella. Amphitrichous, so that means that you would have flagella at each end. We don't have a good image of that, but you would have one flagella at one end and one flagella at another end. That's amphi. Remember, amphi means both. And then the peri, where you have it all over. So this is peritrichous. Here's some other ones that have them just sort of randomly all over. All right, so how are our cells going to move? They're going to move based on reaction to chemical signals or light. So if you're an organism that light affects you, maybe you're photosynthetic, then you might want to move towards light if you sense that there's light near you. But a better example for our chemotaxis, so moving in response to chemicals, if I put a plate of freshly baked cookies up here, and you will be able to smell them, probably from out of the room. If you were to close your eyes, you might be able to sniff out where they were just by smelling. And those smells that we get from the cookie smell, that's a chemical, right? And our nose is just able to sense the chemical in the air and pick it up. They're the same way. They might pick up chemicals for nutrients nearby and want to move near them. Another example might be if I put a plate of literal dog poop up here, you might want to move away from that. So you'd sense the chemicals in the air and want to move further away from those chemicals. And they're the same way. They sense something that they perceive as being dangerous, harmful, toxic, or whatever. They might want to move away from that. So literally just reacting to chemicals. So positive chemotaxis, literally if you can imagine, a positive response to it, they want to move towards it. Negative, want to move away from it. And a phototaxis, reacting to the light. So these guys, like I said, their flagella either rotate clockwise or counterclockwise. And then basically, if they're rotating at all, they're moving in a straight line. When they switch the rotation in a different direction, it causes their cell to tumble. So that will change their direction. They can sort of control that. They don't have very particular control over it. So they'll just run and tumble until they start feeling like they're going in a certain direction and kind of keep that up. Obviously, the more flagella you have, the more you can kind of control that. And so there would be that going on. But of course, the more flagella that you have, the more energy you're going to be putting into running those flagella. So that's another thing to think about. But runs, usually you're spinning the flagella. A tumble is when you reverse the direction and just causes you to suddenly, it's just like if you were to be driving your car and you're in first gear and you just suddenly shifted it into reverse. Your car would not like that. Obviously, your transmission would not forgive you. But you can imagine like your car would just freak out for a second and might try to reverse a little bit, and then you would try to go forward a little bit until, also obviously you have turning going on here as well. But yeah, so that's literally how they're doing it, run and tumble. And so you can imagine if there's nothing going on that could cause them to want to move in a particular direction, they're just randomly kind of running and tumbling, just kind of getting a feel for their environment, randomly experiencing it or whatever. But once they sense something that is a positive or a negative attractant or repellent, then they will have a little bit more control over their runs and tumbles and how they're using it to move in a certain direction. But you can see in this second image here, it's not entirely controlled. We're moving generally in a direction, but each movement might not be a specific direction. Does that make sense? So yeah, it's not very controlled. All right, moving on to our other appendages, our pili and our fimbriae. The pilus, which is the single, or the pili, we also call these the sex pili. I'm going to get to that in a moment. Where the pili can be used for adhesion, they're never used for locomotion. And the fimbriae, that's what their purpose is, adhesion. Again, not locomotion. Flagella, locomotion. Fimbriae and pili, attachment. But the pili can actually have more functions than that. So the fimbriae, think of them primarily as being our attachment guys. They look like tiny little hairs, and they have just molecules on them that allow them to attach to surfaces. So little bristle-like hairs, and they're usually made of protein that allows for attachment to surfaces. E. coli, some species of E. coli have sophisticated fimbriae that allow them to attach to certain epithelial cells, including those that are in your urinary tract. So those that have these advanced fimbriae can attach to the epithelial cells in your urinary tract, whereas other bacteria might not be able to. So instead of urine flushing them out, they can stay attached to the epithelial cells of your urinary tract. 80% of urinary tract infections are caused by E. coli in your own body. Okay. All right, we'll talk about that later. Okay, so pili, these guys, they can be used for attachment, but their biggest function, probably the one we want to focus more on for this class, is that they can form these channels between cells. And so yeah, it's made up of this pilin or pilin protein. I don't care how you say it, as long as you can recognize it on the test. So these are used in conjugation. So I'm sure you guys have heard about like prisons having conjugal visits. So this is that. Okay, so think of when I say conjugation or conjugal or something like that. In this context, you can think sex, because these are actually called sex pili. And what do these allow for? DNA transfer between these bacteria. Now, if one bacteria has antibiotic resistance genes, it can make a channel to the next bacteria next to it, make a copy of those genes, and just transfer it on over to its buddy over there. So we are talking about swapping DNA. So it is sort of like conjugation, like we would typically think of it. So it's our sex pili. They are swapping DNA through those channels. All right, we can also have those surface coatings. We talked about making biofilm and making the sticky stuff on our surface of our cells that allow us to stick together. This is called a glycocalyx. And again, if you hear glyco, I always want you guys to think about sugars. So glycocalyx is usually a repeating polysaccharide unit. It may have some protein involved in there, but definitely think about the sugars, right? And we can have it in a loosely formed layer, which is nice gooey, like really loosely associated, or we can have capsules, which are more tightly bound, nice neat coating around it. Either way, these are pretty sticky. These are usually associated with adherence to surfaces, but our capsule actually has more function than that. So here's some images of encapsulated. We're actually going to do a stain very similar to this one. We have our background that's stained here, so we're using a negative stain to stain the background, a positive stain to stain the cells. And here, this white area here did not stain. That's because the capsules of these guys tend to be nonpolar and they don't stain very well. They are just going to repel stains. And so you can see here that white halo, that's that unstained area. That represents that sticky capsule that's around these cells. We can actually visualize it with certain kinds of stains. And you guys will be seeing that in the lab. Here, down here in this image here, we have a species of bacteria that isn't producing a capsule. And then we have caused it to produce a capsule in the next image. You can see the difference. That gooiness on the surface is even seen in the actual growth on the plate. I'll say this, most of the bacteria that we're going to be dealing with, most of them will have capsules in the lab, because they aren't just useful for sticking to surfaces. But you can imagine if you were a pathogen, that's already a useful step to have, something that will allow you to stick to surfaces in your host. But they're also really helpful. Having a nice thick sticky coating on the outside makes it hard for your white blood cells that are defending your body to grab on and eat these guys up. So it's like a sticky, slippery kind of surface it can't really grab on well to. So your cells are trying to grab on to bacteria, and if it's just bacteria, it might be able to grab on easily to it. But if it's got sticky, slimy coating, it can't grab on very well. So having a capsule helps prevent against something called phagocytosis. So phago means eating, and cyto means cell. So we're talking about cells that eat up other cells. So that's our white blood cells. That's what they're going to do. That's how they protect us. They'll eat up anything they see as a bad guy and then digest it down. They're also going to eat up debris, dead cells and stuff like that in your body and digest those down too. But this is how they work in their immune system. We'll get into the details of how that works as we go into unit 3. But for now, we're going to talk about phagocytosis a lot. So at least understanding that concept when I start talking about phagocytosis, and if I put on a test, what are capsules, good at preventing? And if one of the choices is phagocytosis, it might be helpful to know what that is. This is when your white blood cells eat up bad guys. And obviously, capsules are pretty good for helping out with making biofilms. They give you that nice sticky coating to stick onto surfaces as well as to other bacteria and build up one another. All right. The cell envelope. So the cell envelope refers to the entire outside structure of these organisms. This will include, like we've talked about with all cells have a membrane around them. All cells do. Even our Archaea, which they don't have phospholipids, they have some other weirdness that you don't need to know about, but they all have a membrane of some kind. Okay, so with bacteria and with all eukaryotes, we're talking about a phospholipid bilayer. Okay. So that's our cell membrane, and our cell membrane we can also call a cytoplasmic membrane. Cytoplasm is the goo inside of your cells. So it's the goo that supports everything that's going on in your cells. It's water-based. We'll have some electrolytes in there. And then whatever proteins and other molecules are going on inside of the cell, all contained in the cytoplasm. The cytoplasmic membrane, therefore, is the membrane that contains all that goo. And it's a membrane that's directly in contact with that goo, because some of these bacteria have two membranes. Okay, so the cytoplasmic membrane, which will always be the inner membrane, the one containing the goo, and they might have an outer membrane on the outside. Most bacteria also have a cell wall, not all of them, but most. And that cell wall is either thin or thick if it has one. And it's always going to be made up of, in bacteria, it's always made up of something called peptidoglycan. But all of these are going to work together as a single protective unit. And these are actually going to be our defining factors when we start talking about the differences between our bacteria, gram-positive and gram-negative bacteria. Number 1, in identifying differences between bacteria, the most important first step is determining if your bacteria is gram-positive or gram-negative. And certain antibiotics will work better for one or the other. So this is actually really important in determining the identities of these guys, because the two most common causes of meningitis, for example, Neisseria meningitidis, which is also known as the meningococcus, and then Streptococcus pneumoniae, which sounds like it causes pneumonia. It is the most common cause of communal pneumonia, but it also is the most common cause of meningitis, bacterial meningitis. And Streptococcus pneumoniae is a gram-positive bacteria, whereas Neisseria meningitidis is a gram-negative bacteria. So already right there, typically if a patient comes into your ER and is presenting with meningitis, the first thing they're going to do is get a CSF sample through lumbar puncture. When they get that spinal tap, you can take that CSF sample and send it to the lab for a gram stain. And that gram stain will tell them literally everything they need to know, just from one gram stain, on how to treat your patient with what antibiotic and how and everything. In the meantime, we're going to throw broad spectrum antibiotics for both gram-positive and negative, and just hope that we're covering our bases, because meningitis can kill so quickly, you don't want to wait for those results. So that can be a problem. Meningitis, by the way, can be caused by bacteria, fungi, and viruses. So you do need to figure out what's causing it. But because bacterial meningitis is considerably more severe than the other two, and can kill much more quickly, we're going to just throw antibiotics at it no matter what, right off the bat. So we can talk about how to tell the differences between what kind of meningitis you might have just from a gram stain. Because from a gram stain, I would actually be able to tell you if it was the meningococcus, the pneumococcus, fungal, or viral, all from just a gram stain. So that's helpful. All right, so what's a gram stain? We're going to talk about this. This is going to be lab exercise 3. We're actually going to be doing gram stain. It's going to be our first step towards identifying our unknown bacteria. But basically, it's a staining procedure. It's a differential stain where the stains react differently to different molecules in your bacterial cells. So gram-positive bacteria have a very, very thick cell wall composed of peptidoglycan. So if I were to draw that, let's see if this will work here. Oh, thank God. Okay. All right. Let's do this. Annotate. Can I draw with this? Yes. Okay. So I had a thick cell wall. And we're going to talk about this, what this means as far as thick cell wall. And then on the inside here, you would have your phospholipid bilayer. Okay, so you have your little heads that are joined here. I'm not going to draw the whole thing, please forgive me, but you get the idea of how this will go. The little tails and the phosphate heads. This is your phospholipid bilayer that makes up your cytoplasmic membrane. So I'll just write that here, cytoplasmic membrane. And then here is the cell wall. This is the gram positive. And the cell wall is made up of something called peptidoglycan. And it says it in the name what it is. Peptides. You guys remember what a peptide is? Short chains of amino acids just linked together. Usually less than 20. Otherwise, you're talking about polypeptides, right? So you have short chains of amino acids linked together, and then glycan gives away another molecule that's in there. You guys remember what that would be? Sugars. So we're just talking about amino acids that are linking together chains of sugars. So we'll have long chains of sugars cross-bridged by our amino acids. I'll show you what it looks like in a moment. That's what peptidoglycan is. And it's like a scaffolding that's just built up to make up these walls. And then we have our cytoplasmic membrane that's on the inside containing all the goo, the cytoplasm. For our gram negatives, they really only have like one layer of peptidoglycan. So a very thin layer of peptidoglycan. Then they'll have their membranes, the cytoplasmic membrane on the inside. I wish I could just copy it, but you know, whatever. And then we'll also have another membrane on the outside. They have an outer membrane. Because they have a very thin cell wall, it just doesn't give them enough structure and support. So the outer membrane just adds into that extra support that would be lacking because we have a thin cell wall. So they're compensating by having that extra outer membrane. So outer, and this is cytoplasmic, the Y. And then this is the wall. That's made up of peptidoglycan. So this is our gram negative. So now you're starting to see these are drastically different cellular envelopes from one another. You're taking an antibiotic that targets the ability of your cell to build peptidoglycan. You could see how a gram-positive bacteria, which depends very heavily on this thick cell wall peptidoglycan, if you're affecting the ability to have that, it's going to affect a gram-positive more than it would a gram negative, because at least the gram negative has another outer membrane to support them. Now, will it affect the gram negative? It will. It's just going to be less effective typically. But those gram positives, they depend on that cell wall. That's actually really important for the cell structure. Their cytoplasmic membrane, that inner membrane that contains the goo, will actually be attached to that cell wall to the point that if you put them in salt, we put our cells in salt, what happens? Our cells shrink down and lose all the water that's in them to the salt around them, right? They have a big, thick cell wall around them. They attach to that. They don't shrink down. They stay attached to that cell wall. It stays nice and rigid. They don't get killed as easily by salt. So when I tell you, gram-positive bacteria include species like Staphylococcus aureus and Staphylococcus epidermidis and other organisms that live on your skin. We already know our skin is salty. Y'all licked skin before? We know that it's salty. You got salt in your sweat and everything. The saltiness they can withstand because of that thick cell wall, giving their cells that extra structure. Gram negatives don't do as well with that. However, gram negatives do well in other environments like the gut. The deep inner colon, gram negatives typically do better. Okay, so there's other factors involved in like their environments that they're living in and everything like that. Oh, good, that does that. Cool. Sorry, can't keep that. All right, so here is a better drawing than the one that I was giving you guys. You can see here in this image that we're looking at here, our gram positives. So you can see our gram positive on the left, that purple thick cell wall. That's going to remind you guys that whenever we stain these guys with the gram stain, positive is purple, P, purple. Okay, positive P, purple, positive P, P, P. Okay. Our gram negative, to me, red is a negative color. Traditionally, we think that stop and things like that. It's a negative color. So our gram negatives stain red in the gram stain. We'll talk about how that happens in a little bit. But here we can see here depicted here that thick cell wall with our cytoplasmic inner membrane for our gram positive, which is here on the left. And then our gram negative, we have these two lipid bilayers sandwiching that thin layer of peptidoglycan there. All right, makes sense. There's some other factors that are involved with this. Gram positives also have the structures to support all this. We have built up all these high layers of scaffolding of peptidoglycan in order to support that and to help when we're making copies of these cells, because these guys are constantly making copies of each other, and to help support building that thick layer when we're building new cells. We have lipoteichoic and teichoic acid. All right, so these guys have that. So they both have teichoic in them. All right, gram negatives don't have that. But because they, number one, they have this whole other freaking lipid bilayer on the outside. And they have something else in them called lipopolysaccharides. So this is we can abbreviate this as LPS, lipopolysaccharide. And it tells you what that is too. We've got a lipid component. And then the polysaccharide, you guys know what that is, right? Sugars. So these markers that are on the outside of gram negatives are another thing we could test for. So even if we can't actually see in a microscope that thick cell wall or that thin cell wall, but the chemical test that we're going to do will be able to tell us if it's there or not. But we could also test for things like teichoic acids or LPS. Now, LPS is incredibly important in diseases because LPS, we also know this as something called endotoxin. Endo means inside. And we have toxin, that just means that it causes damage to our cells somehow. It has a toxic effect on our cells, so literally means it's going to be damaging or killing our cells. And endotoxin is in because it's part of the bacteria, in the bacteria, okay? They're not secreting it out into the milieu. It's part of them. And our body reacts to it like a toxin. So if you start getting this in your bloodstream, this can cause toxic shock and things like that immediately. We'll talk about how that works later on in other chapters. But LPS in the gram negative, defining feature. Really important that we know that LPS, the lipopolysaccharide, is the same thing as endotoxin. It's another thing I'm going to bring up a whole lot. Okay. Oh my gosh. So we know that our cell walls are going to help with the shape and the structure of our bacteria, how it reacts to salt and things like that. And we know that they're made up of peptidoglycan. It's basically a scaffolding. Let's see if I have a picture of it. Yes, I do. You don't have to draw it. You don't have to recognize it. But hey, this shows us that it is a scaffolding. These G's and M's. Oh, that's not right. The G's and the M's here stand for different sugars. They're alternating sugar. So here we're talking about N-acetylglucosamine and N-acetylmuramic acid. You don't have to know that, okay? But they usually write them as NAG and NAM. And so that's why they have a G and M here. So NAG and NAM. And they just alternate, G, M, G, M. And then we'll have long strings of GM, GM, GM going on and on and on and on. And cross-linked occasionally by these green branches that we have here. These represent just peptide cross-links. And what I mean by peptide, remember that a peptide is just a sequence of amino acids. And this would be NAM, N-acetylmuramic acid. And we would have linked in between these guys, each of these amino acid chains that we call a peptide. It's just a bridge. This is an amino acid, L-alanine, D-glutamate, each of these is an amino acid. There's different kinds. You don't need to know what they are or what order they're in, but just know that it's amino acid chain linking these sugars to each other across. So we have these chains of sugars cross-linked by these bridges of amino acid chains. Okay, that's our scaffold. You can see how it builds the scaffold when you look at it like big picture wise. So we'll have thick layers of many chains cross-linked together in our gram positive. You might just have one layer in the wall for a gram negative, just one chain. Alrighty. All right. So we already mentioned our gram-positive, thick peptidoglycan layer, multiple layers of those sugar cross-linked with amino acid chain scaffoldings, supported by our teichoic acids that are important for growing that cell wall when we're making new cells, as well as just maintaining that thick wall. Okay. It also gives an acidic charge on the surface. Whereas our gram-negatives, we have a very thin 1 to 3 nanometer thick peptidoglycan layer, often maybe even just one chain of peptidoglycan. It gives it less rigid, but it still has shape. And a lot of that shape of their cell is defined by that outer membrane. And that outer membrane is studded with lipopolysaccharides, LPS, endotoxin. It's all the same thing. So we have greater flexibility in our cell shapes with the gram-negative, but we're more susceptible to being lysed by like salts and things like that. Whatever. Moving on. All right. Our here's lipopolysaccharide. Again, I'm not making you draw it or recognize it or anything, but this is just showing us we have our lipid tail and we have a polysaccharide portion up here. This is also known as endotoxin because it stimulates fear. So think of endotoxin as being like the medical term and lipopolysaccharide being like the science term, okay, or chemical term. We also have some bacterial cells that do not have cell walls, or they have modified cell walls. So to start with, we have the ones that have mycolic acids. These are not typical. Here, we have a thick peptidoglycan wall, but it's not as thick as usual gram-positive. So this will stain gram-positive. But instead of that, just that typical thick peptidoglycan block, they'll have like a partial peptidoglycan. And then on the outside, they'll have these waxes. I'm just going to put, I don't know, some blobs up here. And these are waxes. And this wax is called mycolic acid. It's a type of wax. It's a long chain fatty acid. So it's a fatty acid. It is an acid, but it's a fatty acid. So it's that lipid component that we're way more concerned about here, because that is going to be hydrophobic. This will make them more resistant to certain chemicals, certain dyes and stains. And so we use a special stain to identify if your organism might have this. There's only certain kinds of bacteria that can have this. These are Mycobacterium and Nocardia. So I've told you about this, that it's only two, that it's going to be Mycobacterium and Nocardia. I'm never going to test you on Nocardia. Why? Because Mycobacterium is considerably more important medically, okay? So I'll often test you on Mycobacterium. There's two types of Mycobacterium. Well, there's kind of three that we want to be aware of, but two biggies that cause very big, obvious, you've heard of them types of illness, okay? Tuberculosis and leprosy. Mycobacterium tuberculosis gives it away in its name. And then Mycobacterium leprae, leprosy, okay? But they're always going to be named Mycobacterium something. And if it is called Mycobacterium, it has mycolic acid, okay? I'm going to confuse you guys. There's a special stain that stains this. That's the acid-fast staining procedure. We'll learn this in a lab exercise 4. It is a lipid-soluble stain that we will use that binds specifically to lipids very well. Most cells don't have lipids on the very, very surface, right? We have the charged, heads. But this wax being on the surface of these cells, we can actually stain their surface pretty well if it's there. So automatically, I'm telling you, you can take the sputum of a patient that comes into your ER coughing, have them cough into a cup, sent to the lab, and they can literally do a gram stain on the sputum directly. Not gram stain, excuse me, apologize. They can do an acid-fast stain on that sputum directly and to tell you if there's acid-fast bacteria there. If they are acid-fast, they have mycolic acid. If they have mycolic acid, they must be Mycobacterium or Nocardia. Nocardia isn't associated with medical illness that we care about. But Mycobacterium, if they're coughing up blood and they're already there for coughing and it's acid-fast, what is it going to be? TB. Okay, a third of the world has TB. This is not a small thing we're talking about. All right, very, very commonly done. I used to work in the lab at Integra Southwest. Commonly got this test. Commonly, okay? All right, this is the #1 test they're gonna reach for. It's cheaper to do that acid-fast stain than it is even to do a chest x-ray. So it's really easy to do. Next, we're moving on to mycoplasmas. Great, another myco. These guys don't have mycolic acid. So here's how I'm going to help you tell the difference, and you're probably going to get confused anyway. Mycobacterium just tells you that they're bacteria with mycolic acid, okay? These are mycoplasmas. So if you had to choose, if you had a list, you had to choose between mycobacteria and mycoplasma. The way I remember these guys don't have any cell wall at all. They're pleomorphic. They don't keep a shape. So if you're walking around with a cup of plasma, and most of you guys know what plasma is like, right? It's like a golden yellow color liquid from your blood. We're talking about plasma, right? You donate plasma. You know, it's like a golden yellow liquid. You're walking around with a cup of plasma and you spill it on the table. Will it keep its shape? No, right? It's liquid. It's like water. It's mostly water. So same with these guys. Plasma, think like you're dumping a cup of plasma. It doesn't keep its shape. Why doesn't it keep its shape? Doesn't have a cell wall, okay? Like our cells, these guys have sterols to help give them some shape and rigidity, but they still don't have a defined shape, so they're pleomorphic. These guys are typically the cause of walking pneumonia, which is also going around right now, FYI. So if you've heard of somebody having walking pneumonia, it's almost always a mycoplasma pneumonia. So we wouldn't use antibiotic that targets a cell wall on these guys, would we? Wouldn't make any sense. All right, so that cytoplasmic membrane that holds all that goo in, it's selectively permeable, which we'll talk more about in chapter 9. So I don't want to get too much into it, but basically what that means is only certain molecules can actually cross that membrane because of the charged heads and the lipid tails. It's just weird to most chemicals. They can't cross, but water can. And that's really important when we start talking about tonicity. So hypertonic, hypotonic, and isotonic. Again, it's a chapter 9 thing. It's important to understand the purpose of that cell membrane is mostly to contain what's in the cell inside and keep what's outside outside. We might need to get things from inside the cell to pass to the outside by secretion. And so there's mechanisms for the cell to deal with that. We don't know about the details, but it exists. So how does the gram stain work? Coming back to our cell envelope, what is the tell between a gram positive and a gram negative? We have to just do the gram stain. And it's actually defined as, we'll define organisms as gram positive and gram negative, literally based on how they stain with the gram stain, okay? So yes, if you're gram positive, typically thick cell wall, peptidoglycan, and that cytoplasmic membrane, and typically gram negative, outer membrane, thin cell wall, inner membrane, typically. But those other guys that we just talked about in the middle might also stain gram positive, gram negative. Okay? Those are the only exceptions. Those are the two we talked about in the middle. Mycobacterium, mycoplasma. Mycobacterium, wax on the outside, they'll stain gram positive. The guys that don't have a cell wall at all, they'll stain gram negative. We'll talk about why. We'll talk about this, okay? Our first step is crystal violet. Crystal violet is obviously tells you what color it's going to be, luckily. It's purple stain. It sticks in that scaffolding of peptidoglycan. So see how we have this basically here, like plinko or something? You're going to stick those crystal violet guys, they get stuck in that scaffolding really easily. They get trapped in there. In the gram negatives, they will stick and hang out there, that first. They'll stick superficially. So you could use this just to stain with a simple stain if you needed to. However, it's the next steps that determine everything. So here, with just crystal violet, see how everybody's purple? Or the next step, we're adding iodine. Iodine is something called a mordant. It's going to fix crystal violet that's been trapped, that's truly trapped. Now, these guys are only superficially trapped, so there's no fixing on our gram negatives. All right, so we do fix it in the gram positive. You can't tell the difference yet in the color though. Okay, so still everybody's purple. The next step, this is the most crucial for sure, is the alcohol step. This is just ethanol, like drinking alcohol. And this is our decolorizer. And yeah, you do need to know what happens with each of these and why we're going in certain steps and everything like that, especially for the lab, but you do need to know it for the lecture too. Because if I were to ask you that, you know, okay, you sent your sample to the lab, they did a gram stain, but they forgot the alcohol step. Uh-oh. And they're telling you it's gram positive. Is that a reliable result? No, right. So ethanol will decolorize it if it isn't fixed. So if the mordant didn't fix it in place, it will decolorize it. You see how gram negatives decolorize here? We're going to wash out all of that crystal violet that didn't fix in place. Now, obviously, we fixed it in place with the iodine in our gram positives. So they'll stay purple. And then we have clear cells now for our gram negatives. And that's really hard to see in the microscope. So our next step is safranin, which is a red dye. This is just our counter stain, so that we can see the ones that are negative. It gives us contrast, right? So this is like a similar step of what we would just have with like a simple stain. So here, that red is not strong enough to be seen past that purple. It doesn't, you won't see it. So you'll see the purple. But our gram negatives will have rinsed the purple out, and so you will see the red. Okay, and so that's how we wind up with that. So now you can see here, if we had mycoplasmas that don't have a cell wall, they're not trapping any of that. So of course they're going to stay in gram negative. And I already told you, just for sake of you understand, that our mycobacteria with the wax on the outside, they're still going to stay in gram positive. That wax also helps trap it in there. Okay, but other than that, our basic, most bacteria, if they are not mycoplasma and not mycobacterium, it's literally in the species name, they're going to be either gram positive or gram negative. Right? You cannot be both. You cannot be both. You are either gram positive or you are gram negative. That has to do with your cellular envelope. And now you can see the huge difference between these two. There's a big difference. And so you want to be aware of that difference. And that stain is what's going to help tell you the difference within 10 minutes. All right. That's a gram stain. We already introduced the cytoplasm. It's the goo that's inside. It's mostly water. It's going to have your sugars, amino acids, and salts, and things like that. We're also going to have other structures that have function inside the cell that are not membrane bound, like ribosomes and other, the cytoskeleton and things like that. So we'll talk about functions of ribosomes. You don't even know them yet. We'll talk about them another time. Bacteria have a chromosome that is always circular. It's a circular strand of DNA. Bacteria have DNA. Archaea have DNA. Eukaryotes have DNA. Okay, they all have DNA. This is a single circular strand of DNA. So keep that in mind, it's circular. Our chromosomes are not circular. They're linear. And we have a lot of them. We have 23 pairs just in humans. So bacterial chromosomes, they'll just have one circle. They don't have it in a nucleus. It just hangs out in a region that they call the nucleoid region. They can also have extra little bits of DNA that are smaller circles of DNA that have non-essential genes. So they don't need these to survive, but they might have extra genes for things like antibiotic resistance that can help them survive better, or making certain toxins and things like that. We actually can hijack plasmids. That's the name of these extra bits of DNA. They're also circles. We can actually hijack them and put our own genes in them and have the organism make the make genes that we want, that we're interested in. Then we have ribosomes. These are made up of mostly RNA and some protein, but the RNA is the most important for us, called ribosomal RNA. And these are the machines that are going to help us make protein. That's all we need to know about them for now. I'm not going to ask about the sizes. We might bring it up later. The fact that bacteria have their own size and eukaryotes have their own size is important later on. I don't need you to know the sizes. Next, we have bacterial endospores. These are structures that bacteria make within their cells that help them go dormant in harsh conditions. So only certain species of bacteria will make endospores, and these are Bacillus and Clostridium. That's in their name. Their name will be Bacillus or something Bacillus, like Lactobacillus or something like that. And then we have Clostridium species. There's only two. Okay. So these guys are always gram positive. that thick peptidoglycan layer actually helps with the hardiness of these guys. Well, they'll create this whole cortex coating around them, and that helps protect us from harsh environments. So they can actually survive being desiccated. They can survive hanging out just without water or food for years. This is like things like anthrax. So we can dry anthrax down, and the endospores can hang out in that dry powder for a very long time to use as bioterrorism agents. In nature, anthrax is something that exists naturally in cattle, and so that can actually be transmitted into the soil and hang out in farm soil for decades. And so you could buy land from somebody if they had cattle on that land, could have put anthrax into that soil. You go to till your soil, it releases it into the air, and now you have anthrax in your lungs. So that's a common way of getting anthrax. Another example would be Clostridium tetani. What do you think that causes? Tetanus, right. So we're talking about these endospores that can hunker down and hang out in soil for very, very long periods of time. You step on a nail, these guys hate air. They cannot stand oxygen, which is why they hunker down when they're in the air. But as soon as they get into that deep tissue, when you step on a nail or something, they're not exposed to oxygen anymore. And now they can start making their toxin that causes your muscles to contract and not be able to relax. And so that's tetanospasmin is the toxin. That's what tetanus is. It doesn't just cause lockjaw, it'll cause you to suffocate because you can't breathe anymore because your diaphragm will seize up. You die from that. Not just lockjaw, that's baby stuff. So these guys have two phases in their life cycles, vegetative and endospore. If they're endospore, they're completely inert. They are not reproducing. They're totally dormant, okay? If they're vegetative, that's whenever they're reproducing and metabolically active. In fact, we'll call all bacteria that are just normal metabolically active bacteria vegetative cells. Only Bacillus and Clostridium species will do the endospores. This process that they go through for making endospores is called sporulation. And germination is the process from going from the spore to the vegetative cell. There's a nice pretty figure for this, right? They can resist pretty much any conditions, and it's really hard. You can't even boil these guys away. You cannot boil endospores away. So anyways, so the cell that's making, so a cell can make an endospore. It cannot make more than one. This is not a reproductive method, all right? It's just transforming your cell. Transformation from vegetative to spore takes 6 to 8 hours, but going back from spore to vegetative only takes 1.5 hours. So from the moment you step on that nail, within 1.5 hours, you could have active reproducing bacteria in your deep wounds. This is a figure that demonstrates going between, I'm not going to go through the steps of all this, be aware of where sporulation is, what a sporangium is, and all of those terms that are associated with this. Okay. Usually, rehydration with water is often the thing that will cause you to undergo germination back into that. So here's some examples. We also have gas gangrene and botulism falling into this category. All right. Archaea, they live in extreme environments. They are not bacteria. They have their own weird composition of their membranes and their walls and everything like that. They don't have peptidoglycan. They have weird membranes, and they live in extreme conditions. There's only one Archaea we're ever going to talk about, and it's one that contributes to dental decay. We'll talk about it when we get to it. These guys produce methane, and they live in really extreme environments, and that's pretty much what we need to know about them. Methanogen is a term for bacteria or Archaea that produce methane gas as a byproduct. Methane is probably a bigger contributor to global warming than carbon dioxide is. A halophile is an organism that lives in high salt. I mean high salt. I don't mean like salt on your skin. I mean like very salty environments. These are extreme halophiles. Things that live in literal salt lakes and salt. Okay. They often produce a red pigment. Then we have psychrophiles. Psychrophiles live in the cold, and then hyperthermophiles that live in extreme high temperatures. We're talking above boiling. Remember, 100 degrees Celsius is boiling. These guys thrive at like temperatures at like 113 degrees Celsius. Tell me that's not crazy. So we'll often group our bacteria and Archaea based on their cellular envelopes. So we've already talked about the guys that have the thick cell wall, the guys that have the thin cell wall with the outer membrane, and the guys that just don't have a cell wall, right? And then we have the Archaea. They just don't have any peptidoglycan at all. And we actually talked about this in the lab a little bit. So I brought up little Joey Mendoza. So gracilicutes. So actually, let's start with firmicutes. This is how I remember these, okay? So for firmicutes, firm because we have that thick cell wall. So those are gram positives, right? My next one, if we already did gram positive, let's go to gram negative. So G for gram, gracillicutes, okay? So we've got our gram positives and our gram negatives taken care of. That's when I bring up Joey Mendoza. So Joey Mendoza, my old boyfriend from middle school. I gotta love Joey. I am writing this for a reason. You know, whatever, this might be what my little notebook looked like, or whatever. I thought he was a cutie. That was a long, long time ago. So Archaea or archaic, old, okay? So Archaea, old. These are our Mendoza cutes. Joey Mendoza was cute. And then we have our tenericutes. Tener literally does actually mean tender. If you think it looks like tender, it actually really does in Latin mean tender. So here we have lack of a cell wall, soft. These are our mycoplasmas, right? That's how you can remember these. I apologize for this. All right, Bergey's manual. Really all you need to know about Bergey's manual is that's a book that has like all of these tests that could possibly be done for your organism, whether it's gram positive, whether it makes endospores, whether it has a capsule, whether it has flagella, whether it grows on these different kinds of media or stains this way or that way. Bergey's manual has all that info in it. It's a book that has all that compiled. It's like actually like a whole compendium of books, and you have to like know which ones to look in for what, and it tells you like which book to go. It's a mess. They have it online now. OSU OKC doesn't have access to it, so I can't actually show you, but you didn't even know what it is, okay? It's just a book. There's a published source for classifying these guys based on their traits. Bacterial species, you know, if we call it E. coli versus Staphylococcus aureus or something like that. You can also have subspecies and strains. We know you can have E. coli that's just normal part of your gut. Most people know that, right? You can have E. coli just in your normal poop. It's normal. It's not the one that causes dysentery that they call, oh, your salad's been contaminated with E. coli. That E. coli is a whole other strain of E. coli called O157H7 that causes dysentery. Dysentery is another word for bloody diarrhea. It's not just something that happens on the Oregon Trail. Anytime you have bloody diarrhea, it's called dysentery. So that's different than normal E. coli that's part of your poop. Because like I said, 80% or more of UTIs are caused by the E. coli in your own gut. Most of you have E. coli already in you. So it matters the species. It matters what is going on the surface of these guys, what molecules they might have that we can use as markers to identify differences. They might be the same species, but we can actually classify those species even further based on other things. So like I said, we're a little over time because of getting this stuff started in the beginning. So guess what? I'm going to go over these. Term for a round cell shape, cocci. Which appendage can bacteria use to move? Flagella. What is chemotaxis? Movements, right, of our bacteria in response to chemical signals. So it could be positive or negative, right? Remember how we're going to rotate those flagella, right? Okay, what is the difference between gram positive and gram negative cells? Right, the thickness of the cell wall, and if you have that extra membrane for the gram negative, don't forget that. We'll go over that again when we get into exercise 3 in the lab, so that'll be a reinforcer for that. And then what is an endospore? Is that dormant, completely dormant structure that bacteria use to hide out from those harsh conditions, right? Completely dormant, and again, we'll talk about that in exercise 7 in the lab. But anyways, all right, I'm assuming you guys have any questions about any of that? I know it's a lot. Apologize for getting late with this computer. If I'd have known it was like this, I would have been here earlier. We're gonna go up to the lab. Please be up there by 11:35-ish at the latest, okay? And we'll get started with the Kahoot by 11:35, and then we're gonna go over lab safety stuff. We shouldn't take the whole time today in lab.